it's a pleasure to introduce JMSchaeffer, and he's going to lead our second panel discussion about novel funding mechanisms. Jim.
Great, thanks, Jamie. First of all, I would like to thank the organizers of this meeting. It's, and I'd like to compliment them. This has been an extremely well-organized, well-presented meeting, at least up to this point. So I congratulate you all. I realize this is a huge amount of work, and you pulled it off, especially for the inaugural event. And second of all, I'd like to thank the sponsors, because that's always helpful to get money coming in and helping support the effort. So today we're going to be talking about novel mechanisms to fund biomedical research. And I will say a few words, a few thoughts I've had about this topic, and then I'm going to ask each of the people on this panel, all 5 of them, to introduce themselves and say a few minutes worth of description of themselves and what their organization does, and specifically how it relates to this topic. After we've gone through that, I'll ask some questions. And we got together and talked before the meeting, obviously, and I asked them to try to be provocative. And in order to stimulate that provocation, we've asked Pete Schultz to join the panel, because he wasn't on it initially. And one of the panelists had an emergency and could not make it, and Rich Heyman is stepping in. So it's slightly altered from what you'll see on your program, but so be it. So I did want to say a few things about non-novel ways to get funding for biomedical research, and I think you're all aware of it. But when you're trying to get funding, it sort of splits into two sections. One is early stage, basic, innovative research, which a lot of the people at Scripps do, and those projects are normally funded by one of 3 mechanisms. The first is the federal government, so that's NIH, NSF, DARPA, and they represent the bulk of the research funding that's going on. Secondly is philanthropic people who are donating, who are giving money philanthropically, and that very often goes to early stage research. And third, what's coming up more recently has been state-funded initiatives. We heard about CIRM, which started back in 2004. It was a very large amount of money, $3 billion, and it funds more than just early stage, but it clearly is doing a lot to help support early stage research in stem cell research in the state of California. And not to be outdone, 3 years after that was initiated, there was an organization set up called CPRIT, which is Cancer Prevention and Research Institute of Texas. So the Texas state put in $3 billion in a very similar format. So those are the major ways that early stage research is funded. And then later stage, and I think of Hugh Rosen when I'm saying this, but what Hugh talked about with his program in S1P1 program that was very successful. It started out being funded by the NIH, and he was Hugh was good and lucky, and he and his colleagues were able to go from early stage research, which was funded by the NIH, right to where they had proof of concept compounds that were ready, that could be shopped out to the venture community and to pharma, and that's what he did. He went to venture. The problem for a lot of people is that gap in between, when you've been doing research and you need more help, translational medicine, to translational research, and that valley of death is the problem, and that's what a lot of people on this panel have been thinking about, and I'd like to probe it. So I think what we'll do is go down from my right, starting with Dan Burgess, and ask each of you to introduce yourself and say something about your organization.
So I'm Dan Burgess. I am a venture partner with SV Health Investors. I've been doing that for, I guess, 5-6 years now, and prior to that I spent about 25 years on the operating side in a variety of biotech companies here in San Diego in a variety of therapeutic areas. SV, as you may know, is a kind of a traditional venture fund that's been around since the early 1990s. We're on our 6th or 7th fund at this point. And we do kind of traditional VC types of investing, and I'm more heavily involved in that piece of it. But a couple of years ago, an initiative by the British government focused on Alzheimer's disease kind of led to a somewhat unusual fund that we have that is the reason I think Jim wanted us to participate, called the Dementia Discovery Fund. So the UK government said we see this huge sort of freight train coming of diseases of aging, particularly related to the brain, eating up enormous resources, and we've got nothing out there to treat this. And so recognizing that, you know, we don't have a lot of good options, we need to do something to kind of spur that along. So they sort of asked for proposals from other VC firms to manage a fund that they seeded initially, but the fund has grown well beyond that here over the last couple of years, to really focus on what we're calling age-related diseases of the brain, primarily Alzheimer's disease, Parkinson's, and to a certain degree ALS. And we focus on essentially discovery through phase 1, trying to get that initial focus of dollars into areas that's hopefully going to be able to produce something new and exciting. We specifically are not doing amyloid-related therapies and don't do a whole lot in the tau area either. But that's kind of the crux of what we're trying to do. We've got an interesting set of investors that includes a variety of big pharma companies that have CNS efforts, as well as Bill Gates personally, AARP, the NFL Players Association, and a couple of other interesting investors who have an interest in seeing our brains preserved. So we can talk more about that as we go along.
Hi, Kathy Richmond, Director of the Paul J. Allen Frontiers Group. It's part of the Allen Institute. So many of you may know the Allen Institute because of its work for the last 15 years of the Institute for Brain Science and the community tools that it's provided with the gene expression atlases. It's evolved greatly since then. So in the last 15 years, it's grown to be a 500-employee, $150 million annual operating budget organization that has 4 business units, one of which is the Allen Institute of Brain Science. But in the intervening time, there's also been the Allen Institute of Cell Science that was launched about 3 or 4 years ago. In December, the Allen Institute for Immunology, and additionally, the Paul J. Allen Frontiers Group. So whereas the institutes have a very mission-driven team science, big science, open science mandate, the Frontiers Group is the external face, and we're really looking at funding pioneering frontier research ideas. We do this both through the Allen Distinguished Investigator Awards, which are 3 years, $1.5 million, for both basic science to translational work. We also have partnerships that we do. We have a recent one with American Heart Association for $43 million to look at both brain health and cognitive impairment. And many of those awardees have translational aspects to their projects. And one of the things that we're excited about, and it's still an experiment, are our Allen Discovery Centers. Those were started just recently, and they have 2 phases. The first phase is 10 years of funding over 4 years, so $2.5 million a year. And then at that go, no-go decision, there'll be an assessment of both the scientific impact, the momentum that's been gained, as well as a requirement for match funding for the following 10 years. So you'd go for a second phase of $20 million, 10 from our organization, 10 from a variety of possibilities. Because we span over such a diverse area of scientific topics, this could be in the form of VC funding, this could be in the form of in-kind, this could be in the form of NIH grants. It really leaves a door open, and some of our Discovery Center topics, systems modeling of infection, as well as morphogenesis and regeneration, really leaves the door open to see how that experiment will play out and what kind of growth that these centers will have. So I'm eager to tell you more about that.
Rich Heyman, scientist by training. I moved here 30 years ago, and I did my postdoc at the Salk Institute, and over the last 20 plus years, I've mostly been on the operational side of biotech companies, on the research side. A few years ago, I morphed a little into helping startups. I spend a certain amount of my time as a venture partner at Arch, and I'm also vice chairman on the board at the Salk Institute, and Pete asked me, I guess about a year ago, to join the board of overseers here at Scripps. So I've kind of balanced my time between startups, the academic institutions, and a venture partner at Arch.
And I'm Barbara Sosnowski, and I'm working at Pfizer. And I've been there for 9 years. Prior to joining Pfizer, I was in the biotech world here in San Diego, and Rich and I were actually postdocs together not at the same lab, but at the Salk at the same time. So we've known each other for a long time, and one of the things that I've always been involved with has been biotech company, basically from startups all the way to a 60-person company. I at one time was VP of R&D, and then I became CEO of a company. Sold that, thankfully, made some money, not much, but when I think one thing that happened after that was that when Pfizer called, that was in 2010, they wanted to set up a new unit that would be a scouting function and that would have the understanding of what it takes to take something from bench, all the way from idea to bench, to bedside. And it's really a novel group. We're very nimble. There's been some recent changes within Pfizer that now I oversee all 5 therapeutic areas within Pfizer, so for scouting purposes. So oncology, inflammation, immunology, rare diseases, vaccines, and metabolic disorders, internal medicine. And so the scouting team that is worldwide scouts for all of these opportunities that go from discovery to candidate stage. And so one of the things we do is we work with academics, we talk to the TTOs, we go to young biotech companies, and we really try to identify new opportunities. And we scout for both the research units, as well as we scout for the Centers for Therapeutic Innovation, which I'll be happy to talk a little bit more about, as well as for Pfizer Ventures. So if we're looking to build a new startup or do a Series A, we can also do that scouting capabilities as well. So my team does all the diligence, and we bring in the diligence teams, and we assess everything scientifically. We pair up with BD. We don't do transactions, and we've formed together a nice team that allows us to move forward or to have quick kills. That's the other thing we do. And we recommend when we do kill a project, we tell you why we killed it and why we're not moving forward. So I've been at Pfizer 9 years, and it's been a great experience. It's probably one of the best jobs I've ever had. And it's always exciting and always challenging because I'm faced with new science every single day.
Barbara, before you go on, can you say more about the CTI now?
Sure. So Centers for Therapeutic Innovation was started as a novel way to identify new science. And so it's an outreach into the academic environment. It started in 2010. The first site was UCSF, and we were all antibody focused. So every modality would have been an antibody coming out of that. As we made scientific hubs within New York, Boston, area Cambridge, San Francisco, and San Diego. And so we have these scientific hubs. And the idea here is to work with the academics in the regional areas, and then partner with scientists within Pfizer. So that together you use the talents from the academic, and you bring together the talents of Pfizer in the development process, so that you can marry those and you can actually bring things to the clinic faster. So that's the whole idea, is bringing things to the clinic faster. We broke away from just antibodies in about 2014-15, where we actually introduced small molecules into the process. And then we branched out from the hubs. And so we no longer are we just dedicated to those 4 areas. We now have 4 sites in Australia. We have a site in South Korea. We have sites in the UK. And so we're expanding that basis. So one of the biggest things when it's first started, there were only 2 scouts to scout for all of the 4 hubs. And then as we branched out into plus 30 sites, we now have 16 scouts that are all based around the world. And so they're helping us bridge that, talking to different sites and identifying the new opportunities. So it's really advanced. We've learned and we've changed and then we modify. So it's really kind of evolved from the 2010 phenotype to what it is today.
Thank you. So Pete, I don't think you need to introduce yourself at this point, so maybe we'll just start out right with questions. Is that all right with you?
Yep.
Good. So this morning after you spoke, I asked you whether, I enjoyed your talk. I thought you explained things very well. It was very impressive. And I asked you, it sounds so impressive and so important. The question is it scalable? And you're sitting right, almost very close to a guy who's at Salk, or who's related to Salk. Could they do the same thing, or could you guys interact with and provide a way to interact better?
Yeah, I think to build what we did, it was a very stepwise process, and as you know, it was started because of you, myself, and Peter Kim's insights. Now that we're moving further along and we're getting more resources, we actually think it would be a really interesting idea to form partnerships with other academic institutions that have very interesting opportunities. So we're talking with folks at Dana-Farber, for example, about protein degraders and so forth and so on. So I think a lot of people are asking us whether we can expand our model, not simply by forming a close relationship between Calver and Scripps, but now forming a closer relationship with other academic labs that have great biology and where we can do the translation part. And in fact, that's what we do with the Gates. The Gates Foundation acts as a catalyst to introduce us to a huge number of other people, and we have a lot of collaborations. So I think that's on our horizon, and I think it's it's really interesting direction, because the idea that you can be an expert at all of the disease biology and do the translational piece is really naive. So you need to partner. Okay, so that's our fundamental thinking. I think what's tougher though in scaling the model is you become resource limited. Okay, especially as you try and move molecules into phase 1. Okay, if you partner those at an early stage, the value you get out of those is relatively minimal, and it doesn't allow you to create a self-sustaining mechanism. You have to depend on NIH and other sources of funding. So that's, I think, the challenge in scaling this. So we've been working with foundations, and as I said, we formed this primer fund. That was an interesting experiment. The idea is people who want to impact the development of new medicines contribute to the fund. It's charitable. You give them their money back plus a modest return, and they can use it for another purpose. Okay. So I talked to a foundation that was going to give a grant to the Lincoln Center, and they said, well, if we give it to you, you might make a medicine for metastatic prostate cancer, and then you'll give us back the money, and we can give it to the Lincoln Center, and yeah, that's it, okay. But it's been a real challenge, because people either see it, people are in the world are used to either making investments where they expect a large return if it's successful, or they're used to giving gifts. So we fell into this middle ground that the Gates Foundation is used to operating in, but not that many other people. So that was a real learning experience in creating that fund, which we thought was a clever idea. I think the other thing that's difficult is, even though you've heard about a lot of programs, what I spend most of my time trying to connect us with people. There are a huge number of people, foundations, individuals, who really want to have an impact on human disease through their generosity. And they don't quite know how to do it, okay? We think for some things they're interested in, we're really good, but it's ships passing in the night. So how do you make a connection with people? Because you see, as academics, some people are being incredibly generous, but they're naive. And I once heard Bill Gates give a talk where he's like, I wasted $15 billion figuring out how to do this, okay? Because he's great at software, but wasn't so great at drug discovery, okay? So I think that's another huge challenge I see, is the people who are doing the science connecting with the people who want to impact the medicine And what's the best way to make that connection? I think that's a huge challenge too. So how do you get the resources to scale it, Jim? I think that's a huge problem.
Yeah. So speaking of difficult challenges, right to my right is a guy who's focused on Alzheimer's, or at least one of the funds is focused on Alzheimer's. So it's almost been 0 successes, and you guys are eliminating some of the targets that other people are still going after. How much stomach do you have for risk? and it's I think obviously if you hit it, it's going to be a huge win. If you guys develop it, if somebody you're funding develops a drug to treat Alzheimer's, it's a huge win. But is 15 years long enough? It says the fund is a 15-year fund.
Yeah, I mean, I think it's a very fair question. I mean, I think, we get asked sometimes, is it a philanthropic fund? Is it a venture fund? And much like the issues that were just discussed, I think, we've evolved a bit as well. But I think, the way we try and position ourselves is applying traditional venture capital metrics to how we're looking at new investments. We use venture capital type diligence, all those sorts of things. And we've in fact morphed away from doing some of the kind of early discovery projects that we thought we were going to be doing into companies or into areas where we think we can actually get a company out of this within a year. We'll do a little bit of incubation. We'll do some seed stage stuff, but we do a lot between kind of the seed and Series A stage where we've got, we think a tractable technology that we can actually get into the clinic in a reasonably short period of time. In terms of targets, it's not so much that we don't think amyloid and tau have no hope. It's just that there are already a number of clinical bets on those. And so we're taking the view that let's see how those play out, and we're actually focused on things like neuroinflammation, mitochondrial function, so on, that we think may actually be complementary to those types of approaches or work on their own, and as a way of kind of seeing if we can kind of move those along while these other things are going forward.
Because when I saw your website, I think it was pretty recently, it's a recent website, it said there were 4 major areas you're looking at now. And I was just wondering, is that closing the door on anything else that's really novel, that's so novel that nobody even knows about?
No, I mean, we will look at all sorts of things. We just try and give folks a guidepost. And it's as much as anything to let folks know that, don't send us the 18th amyloid type approach that is out there. But we've funded some rather unique approaches so far.
Yeah. So, Kathy, I had a couple questions for you. I need some education on this, but you guys fund early stage innovative projects. Does the organization also support translational research and aid in the advancement of developing programs?
So we do have a really broad portfolio, and it does cover a lot of work done in the basic sciences. But an example of one of the projects that we just funded this last fall is a project out of Fred Hutch looking at a new approach to immunotherapy, where instead of removing the cells and engineering them outside of the body and putting them back in, it's actually doing this engineering in vivo. And the funds that were requested or that were needed were to scale up the reagents and do the large animal testing because the proof of concept had been done. So that's an example of the type of work that we're looking at funding where we can make that difference. But it is something that it's wonderful to hear of all of these other models as well, because it is an exciting area.
So are there potential synergies between what you guys are doing and, for instance, DDF, which I think you guys have a big interest in Alzheimer's too?
No, we do. We do a lot of funding in that area, and in fact, that is one of the things that we look at when we're looking at funding, particularly people who are looking at translational projects, what kind of experience they have, what kind of infrastructure they can leverage, and definitely looking at these other models and thinking of partnerships or thinking about investing in these funds is something that's on our radar, because you need more shots on goal, and I think that's exactly what you were saying. Because obviously there's been a great investment to date, and we've learned a lot. It just hasn't translated to interventions or therapeutics.
Okay. Rich, we haven't asked you any questions, but since Rich has just joined the panel. What are you thinking about all the, what's your involvement at Salk as far as looking for novel ways to fund programs there?
Yeah.
Do you have a role in that?
Well, I think I'm clearly involved in kind of helping to think about the strategy from a funding point of view. And I think to the panel discussion here, whoops, maybe I'll make two quick comments. One, over time, it's very, very important that we do think about different models. And I think Trevor hit on that at lunchtime. And, you know, we can come back in a moment to what's going on here at Scripps and Caliber. But if you look at the history of, and I'm pretty familiar with the funding cycle at the Salk, for example, you know, 10 plus years ago or more, most of the money was coming from NSF and NIH money. And if you fast forward 10 or 15 years now, obviously there's 3 or 4 buckets. You know, the buckets are still government money. There's a lot of foundation money that's coming in. And there's a lot of philanthropy. And the budget keeps going up, but the balance between those, you know, that pie changes over time. And then I think you have other newer models like what Pete is thinking about here, more on the translational side. So I think as we think about both funding and just models in general, I think it's very, very important that we are realistic and look in the mirror and say, what are the different groups really good at? And to be honest, what are they really bad at or not good at? And to be provocative, right? If you put buckets in what I'll call straight academia, you know, I'll maybe make a comment on each one. I think they're very, very good at basic research. I think most academic centers don't have a clue about really what drug discovery is all about. I think the group here is the complete exception to the rule, and I think Chris pointed that out this morning. There are many aspects of both creative science, but also execution, and I think you heard throughout the entire morning, the group here can do that very, very well, and Pete's trying to build a new model. So I give them a ton of credit for that. Whereas in most cases, when academics say they want to do drug discovery, I think they understand the early stuff, but they don't understand the execution piece. Now, there are examples, you know, where there's been tremendous success, and, you know, Hugh talked about that, and I had the opportunity to work with a bunch of academics on the prostate cancer side where the actual compound came from an academic lab and it ultimately got approved for prostate cancer. But I think there are things that academia does well and things that they don't do well. On the biotech side, I think they can be creative. They can, you know, live or die by their, you know, projects. But when you think about perhaps where they're not so good, late stage, big phase 3 or phase 4 clinical trials, certain aspects of commercialization where it requires tremendous infrastructure, regulatory issues, that's probably where they often stumble. And therefore, you know, big pharma plays a big role. And then if I jump all the way to the pharma side, I'd say when you think about the budget that pharma has for R&D and their productivity, it's a failure. It's a complete failure. And therefore, I think they do some things very, very well. But when you look at their budgets relative to biotech budgets or academic budgets, it really doesn't, it's not working from my perspective. And I think most senior people in big pharma would agree as well.
Yes, you're sitting right next to Barbara. So, but no, and I think.
Well, but that's where the CTI, for example, is a great creative model for taking advantage of what big pharma is, and that's what Barb talked about,
yes, and
how they interface with either academics or biotech.
I completely agree, Rich. So, Barbara, you've been left alone so far, but and I sent you this question. I'll read it to you. Just I want to hear your response. The CTI describes, and this is in quotes, authentic collaboration involving shared decision-making and alignment of incentives. And then I said, this doesn't sound like the pharma industry I know. So I'm sure you have some questions, some remembrances or stories you could tell of clashes that you've had. Because I can't believe that academics and pharma, once they've been working together for a year or so, don't have some headbutting.
Well, I think, well, first of all, we spend a lot of time with academics. Just we have office hours at the sites, so we don't just come in twice a year and, you know, talk to everybody in the tech transfer offices. We actually work with them. We have science days where we get to understand what's going on. We do have a call for proposal that is twice a year. That goes out to at least the 35 different academic sites. That call for proposals is actually really an innovative science, so looking at nucleic acid sensing, DDR, repeat expansion disease, things that are going to align with our research unit. So one of the key things is that CTI has to, the whole thrust of it has to align with the research unit, because as this grows and develops, at one point it's got to hop into the research unit for further development. So there's got to be an alignment between what's done at CTI, which is at higher risk, to what's actually done in the research unit, and then carried on one step further into the business unit where you're going to have phase 3 done. So there's a good alignment between what we start on versus what we finish on. So with those call for proposals, we do get things that just don't align at all. And we've had some discussions with either the TTOs or the academics about that this really is not a good fit, and why isn't it a good fit? But if it's right on the edge, we have these things called abbreviated statements of work, where we fund something for a year. And it's not a full-blown proposal, but we say, let's try to figure out if this is going to work and can move on into a proposal. And if it can, then we move it that way. If we can't, we just, we try to do a quick kill. So I was just going to say, and then after we, so there, just to give you some metrics, because we keep metrics on everything, there's been over 1,000 pre-proposals reviewed for the CTI since its inception in 2010, of which less than 10% of those, so 83 of those have gone on to full proposals. So the idea is you get all of these pre-proposals, which are a 2-page non-confidential document, and then those are reviewed by both the research unit as well as members within my team, as well as in the CTI leads. And we decide on which ones then to take forward into a full proposal. So then we contact the TTOs as well as the academics, and we work with them together to write the next full proposal. And that's where there's a lot of teaching that sometimes when Rich talked about people don't know how to do drug development, it's really happens that people don't know how to do drug development. And but at the same time, you can teach, we can work together to understand the biology, the mechanism of action. Why are you doing this assay, and things like that, where we are asking the questions, and then we have to listen to them. So it's really a two-way street. And then from those full proposals, we have now in 2019, we have three programs in phase 2, late phase 2, and 2 programs in phase 1. So it's really progressed into, we're actually now seeing from the inception, we're now seeing the fruits of the labor that was the whole reason to actually start this. So yes, there are contentious moments sometimes, but those are few and far between. Most people want to see their babies grow, right? They want to see they don't want to get it killed. And it's when we exited from neuroscience, that was probably the hardest thing, because we exited the footprint within Pfizer. We didn't exit the equity investments, and we still invest in novel sciences, in neuroscience like neuroinflammation, but we had no laboratory to develop this further in a research unit. So everything that was in CTI that was neuroscience focused, we had to shut down, and that was done strategically for strategic purposes, not for academic, not because of science. So
based on the metrics, this has been considered successful.
Yeah.
Are they going to expand it? And the question, I guess, was alluded to earlier. Pharma has a huge amount of money. They're really good at late stage. They could maybe be good at early stage, but they'll never be able to compete with the Salk, Scripps, and the thousands of other labs out there. So should pharma really just drop early stage discovery and leave that to other people? I'll ask that of all of you guys.
Personally, I think not. And I think we're being asked to look at 50% outside, do work 50% outside, and do 50% work inside. And a lot of that's a blend, because we do have other funding vehicles that are very novel. There's this emerging science fund where we're spending, part of the group that I oversee, a ring fence part of money, we work with a research unit who then has identified an academic, and this is technology that we're not, or assets that we're thinking about 10 years down the road, not that's going to fulfill our portfolio priorities right now. So this emerging science fund is supposed to be a quick kill, and we should be developing these experiments to understand, to identify new science. So without those kinds of vehicles, I think you're going to have a harder time getting NIH support, and I think it's going to be a partnership that's really going to be, I think, the key for that.
Okay, Dan, you have a comment?
Yeah, I mean, as somebody who spent kind of the first 25 years of my career kind of pitching science or helping pitch science to big pharma to try and get them to buy it, it's interesting because I think pharma has to have the internal R capabilities to be able to evaluate and validate. I think that's really important. But I think what you found over and over again is that the bar that was set for internally licensed compounds or internally developed compounds versus those that were externally brought in is completely different. And I think even pharma has put out some statistics where if you look at compounds that were going into phase 2 or drugs that were going into phase 2, the hit rate or the success rate of those that were internally developed was something like 1 in 3, whereas those that were in-licensed was something like 2 in 3. And it just kind of shows that the bar that bringing compounds in for is being held to is much higher. And I think if that changes a little bit, and so it really is more equivalent, then I think the whole thing works. And again, I do think the pharma companies have to retain that expertise in-house to be able to successfully evaluate these things.
Does anybody else on the panel have a comment on this?
Yeah, we ran that experiment. So when we started GNF, it was supposed to be a genomics institute and do genomics research, and we built these large siRNA libraries, a huge forward genetics screen and chemical biology cell-based screens and the whole thing. And we spent a lot of time building research tools to probe the genome, okay? And we had the highest throughput. I had an engineering group of 50 people, okay, building tools. And what you realize pretty soon, even with those tools and the rate and the resources you had to do money, you couldn't compete with the academic sector, okay? It was just, even with those tools, there are all of these brilliant people out there and postdocs and graduate students working for a fraction of what you pay your people, okay? And so we actually decided to take those tools and turn them into drug discovery tools and instead start collaborating with the rest of the world, okay? And looking for great science and saying we could help translate that science into drugs. And the latter was far, far, far more productive than the former. And I think the point is well taken that you need people who really understand the research world to evaluate opportunities. But what I saw is the people who were involved in drug discovery read the journals voraciously, okay, because they were always looking for interesting new things to look at. So they had a really good understanding of basic research, but they were really good at drug discovery. And it was like, why would you take a great basketball team and make them play football, and take a great football team and make them play basketball, okay? It just doesn't make sense. So I actually think that, like I think Rich said, academics do research really well, take a lot of risk, and you have a lot of bright young people starting their careers, and they have to do something important or their careers end. And you have pharma and biotech that know how to do the latter. And so I think figuring out how to make them work together effectively and take advantage of their complementary expertise is a far better way to spend our energy than each side trying to reproduce what the other does, quite frankly.
And I think, if you look at the last 20, 25 years, that's already proven itself, right? I mean, the wave of, Pete talked about genomics, but when you think about the early wave of genomic companies, most of them were really failures from a drug discovery point of view. They might have learned a lot of information about genomics, but when they tried to pivot into drug discovery, they almost all failed. In part, they didn't have that mindset or that infrastructure. When academics say that they can do drug discovery, most of the time they don't even understand the pieces of drug discovery. And I think, when you go to big pharma, in many cases, decision-making becomes incredibly complex, problematic, and you don't live and die, by the cycle time of those drug discovery programs. And so I think what is important for all of us is to say, what are we good at? How are we going to get it funded? And how are we going to create new models of pharma relationships with academics, biotech to biotech relationships? What's the right funding vehicles from a venture point of view as well? And, if you pick up, the life science venture, world, you have sometimes companies that are bled money, here's $1 million, get to this point, here's another $1 million, get to this point. And on the far extreme now, you have some of these absolute mega rounds where companies in their Series A on ideas are raising $100 or $200 million. And to me, that's kind of crazy as well. So there has to be a happy medium for doing what you're good at and not necessarily, just raising so much money that all of a sudden you become almost a mini pharma and you don't really have that technology base and decision-making mode.
Jim, I think the other thing that I learned that was a tough lesson is a lot of academics start out doing drug discovery from a neat idea, okay, in pathway or protein to a disease. But what you realize is you probably ought to be working backwards from the disease to the best way to do it, because all of the complexities of the clinical trial and patient recruitment and differentiation and so forth and so on, when you start thinking about that 2 or 3 years into a project. So I think connecting the earlier stage research academics with people who really understand the clinical need and how to test the molecules is something we haven't done well either.
Yes.
So we've been looking for different mechanisms, funding mechanisms, to try, and that's how we rolled out this Allen Discovery Center model. And given the models that all of you are working with and promoting, are there other ways that philanthropists can plug in besides necessarily partnering or funding? Are there ways that we can structure our funding to make it more catalytic, synergistic, dovetailing with your efforts? Or not?
So, a classic one for us was when we were in CF. We actually worked with the CF Foundation and partnered with them. And then when we actually got out, we exited CF and we, gave that remaining money back. But we routinely, to try to understand the disease and what's the needs of from the patient perspective, is we work with foundations a lot. Lupus Foundation is something very keen because we want to be able to design our clinical trials that are actually going to meet the needs of the patient, that are going to be something useful and beneficial to them. Not just that we met this box and that box for our, objective 1 and 2, our secondary endpoints. We want to know that this drug is actually going to have a benefit to these patients. So that's where working with the foundations has been really critical for us, and we need to do more of that, but we are routinely doing that now. In all of our due diligences, as well as when we bring in assets as well into Pfizer and or partner. So.
Okay, so there are about 15 minutes left, 10 or 15 minutes left. If there are any questions from the audience, I'd like to take them now. Melissa? Yeah.
The question is the incentives back to the inventors, and how do you motivate the inventors? I think that's a really good question. The one thing I've kind of learned is now president of Scripps is you really can't tell faculty what they should or shouldn't do. Okay, so if somebody wants to spin out a company, that's quite frankly what they can do. I think what we're starting to see is people like Hugh Rosen and Jeff Kelly and people who have done that now want to do it internally because they want to be involved in progressing their program into the clinic. that creates an issue because, you know, historically, I think academic drug discovery has been somewhat serendipitous. So the participation and the rewards, it's been kind of the outlier rather than the routine. If you now create within an academic institution an effort that's supposed to institutionalize drug discovery so that the teams are routinely putting molecules in the clinic and getting, you know, significant revenues from deals, how do you reward them? That's a little bit trickier problem that we're starting to address now because there are certain rules and regulations about what you can and can't do. And so we have to address that problem because some of the people who do this extraordinarily well and who really are good, it's very likely we can't compete with biotech or pharma to compensate them and what they're doing. So I think a real challenge to our model ultimately is how do we keep people within what we're doing in the nonprofit world and going to the for-profit world. So I think that is a good question, and it's one we're struggling with right now.
But if somebody spins out of Caliber into a company, don't you get a kickback through that? You get some rights retained on that too.
Yeah, but what the issue is, one of the issues is historically if you make an invention in an academic institution, the inventors share in the return. If you make a drug within the Caliber organization, it's not just the medicinal chemist who invented the drug. It's the biologists, the cellular biologists, the people running the animal models, the business people doing the partnerships. So it's this huge team effort. And how do you reward the team versus the inventors is the issue, okay? And if you start not rewarding people who were involved appropriately, it creates a real problem, and it's even bigger problem than that. Because what you don't want to do is reward people based on the success of that program. Because then if you have a really high-risk program and you want to recruit people for that program, it may make it harder because they view this as more partnerable, okay? So I think how we think about doing that in the nonprofit world is something that we're going to face as we move forward with this model.
So Barbara, how about in the profit world? You mentioned that there were a few programs that are in the clinic. How are those people doing and compensated?
Yeah, so CTI is they have a prearranged agreement that if you're involved in that for the antibodies, that they're shared rights. And so because it's a co-development path where Pfizer and the academic come together and make that antibody. So there are shared royalties that go to the inventor and the academic site. For, it's a little bit more complex on the small molecule because we're using our libraries to identify, do all the HTS, high throughput screening. And so that's a little bit more complicated. There are some rights that go back to the university. I'm not sure how the connection goes back to the academic on that one, but there are royalties that come back through the university.
Anyone else on the panel? Yes? Oh, I'm sorry.
Yeah.
Sorry.
Hi, I'm Mary Chamberlain from ADVI. I'm the Global Head of Academic Outreach and External Innovation Scouting. So Barbara, actually my question is for you. How do you guys work your budget for academics? Do you have a centralized budget? Does it come out of the therapeutic areas? Is a little bit of all the above? Do you have the say-so in what gets funded around the CTI? Can you talk to me a little bit more about those pockets of money and what the decision-making process is and how you align with your therapeutic areas?
A lot of meetings. No. What we do is we have a central deals budget, which is actually covers a lot of what we call central deals, and that covers a lot of the traditional type of upfronts and milestones. And anything above a certain threshold is reviewed by the WRD leadership team, so Michael Dolston's group, and then they just make a decision. Sometimes it's subcommittee, sometimes it goes to full review of the 14 or 15 people that are on his leadership team, where it's a little bit, maybe the science a little bit more controversial. So, or it's a little bit more money, things like that. But Usually we have stage gates so that we have that central deals. Now that central deals has to be accountable for the current year's upfronts, as well as accountable for any other deal we did like last year or the year before that might have milestones hitting. So we have to deal with that. CTI has their own budget. So they have, like every other research unit, they have their own budget that allows them to go out and work with the academic sites to bring in, they have a number of proposals that they have to keep the portfolio going. So, you know, we have 10 different early stage opportunities that we're evaluating, and those progress. So we have that ring fence money that comes in through CTI. It gets treated just like another research unit within Pfizer. So all of that is done by a higher level of governance. Michael's on that senior board, and he reviews that with each, I don't even know what it is, PSI, but too many acronyms of Pfizer probably. But so that group is responsible for aligning what the ring-fenced monies are for each of the research areas. So oncology has a huge budget, for example. The next budget is I&I, and then there's different levels of budgets depending on how big the research units are. The other thing that happens is each of the clinical programs is rated against each other across therapeutic areas, so that you're not just funding one that looks good in one area and not funding something that looks even better in a different therapeutic area. So there's a lot of matrix that goes across, which I think is very valuable, but there are different sets of buckets of money, so to speak.
Yes.
Yes, Jim, one of the major obstacles that we see in sourcing philanthropic foundation family office money into translational research is the ability for those funders to feel comfortable about the evaluation, validation of the proposals. And as Peter talked about Bill Gates's $15 billion education, the question is, you know, is there or could there be some kind of an informational repository created that would give funders an independent look at what is being presented to them, so that when they write the checks or don't write the checks, they feel more comfortable than trying to do it by themselves.
Anybody on the panel want to address that?
That's an interesting question. I think there are a lot of groups that peer review proposals for various foundations. I mean, the Gates has the Grand Challenge, and those are peer reviewed and so forth. And, you know, a lot of the proposals we put forth for our various, you know, cancer programs and so forth that were funded by Wellcome Trust were peer reviewed by outside groups. I think part of the challenge there that I perceive is in all of these peer review processes, what you do is put together a panel of experts who really know the area well. And if you're an outsider to that area, I think they question whether you really understand enough of the issues to do it. You know, too often young folks who have really interesting and innovative ideas are not brought into the fold.
You may want to just send any ideas to Pete.
I would just follow up to say that we too take care to pick our review panels very specifically to have people who are embracing of pioneering research and risk and maybe thinking outside the box. It's a lot of times you get what you ask for when you pick these review panels. So it's something that you do take care with, and also just sharing lessons learned from other organizations. We do a lot of discussions with other funders, and that helps quite a bit too for some of these funding areas.
But I will say that I've seen things where people have given huge amounts of money to something that you knew was going to fail. And, you know, that's sad, and it's sad because we didn't get the money, okay, but for the real reason, it's sad if somebody gives a large amount of money to something because they're trying to have a huge impact on a disease for the public benefit, and it fails. It really sours people to other opportunities. So I think when things are, when you know, the funding doesn't go to the best people to get the job done, it has a lot bigger negative impact than simply not spending the money thoughtfully, okay?
I just wanted to make a comment about something you mentioned, and that was whether there has been close communications and work together between two academic institutions, namely Scripps and the Salk Institute. Well, about 50 years ago, when these institutions were quite small, there was indeed a great deal of collaboration between them, plus the University of California School of Medicine, which was just starting up as well. And the meetings that took place were wonderful with the three institutions, and people began to realize that they could help each other. And so we had grant applications sent to the NIH from common people between the various institutions, and it worked beautifully. But then, of course, it grew, and each institution brought in more talent and more talent, so you didn't need to go outside of your institution to get the kind of talent you needed. And then, of course, when translational work came along, it changed a great deal as well. But I just wanted to make out the point of history that in those very early days, there was a tremendous work together between the various organizations.
I appreciate that comment. I think it's probably gotten better in the last few years, but maybe I'm wrong on this mesa.
I think there are a number of us that think we ought to revisit the past because it worked well, and maybe we should start doing that again.
Okay, I think our time is up, I'm afraid. So thank you very much for your attention. Thank you to the panel.